Viruses are minimal life forms without their own metabolism, but they can infect human cells and cause serious illness. By adaptation, viruses can learn to be transmitted more effectively or to escape the human immune system. And even completely new viruses may emerge, posing a dangerous potential.
Viruses can mutate, so sometimes viruses can acquire either an ability to spread faster or the ability to be more dangerous. And so we try to be prepared in the future by studying the viruses that we know, so that we can better respond to viruses that we may not know, or viruses that we know that are changing over time.
The Collaborative Research Centre 1648 addresses newly emerging viruses that have the potential to cause epidemics or even pandemics. Here, basic and clinical scientists with complementary expertise are jointly investigating viruses from the molecular structure to the cellular level, from experimental animal models to human disease, in order to better understand how viruses can infect cells, how they interact with the immune system, and how they can cause disease.
We have experts in virology, molecular biology, structural biology, immunology, and even early drug discovery stages. And together, we can use cutting-edge techniques like AI, structure prediction, cryo-electron microscopy, structural mass spectrometry, and many other techniques, for example, in immunology. And we can combine all of this to better understand virus mechanisms and to prepare new ways of intervention.
Although viruses are tiny and lifeless, they can have a strong impact on the health of individuals and by infecting many people on a whole society. Viruses use their envelope proteins to penetrate cells. They hijack their metabolism, replicate, leave again, and then infect other cells. The infection spreads. Every reproduction cycle could change the properties of a virus, and these changes can turn out to be threatening.
Research into emerging viruses is important because we've seen just recently in the COVID-19 pandemic how the world can be totally affected by just one virus. We have to prepare for these scenarios and understand those viruses better to be better prepared for a next pandemic.
For their work, the scientists at CRC 1648 have access to outstanding technologies and facilities. High expertise and excellent infrastructure allow state-of-the-art virological research. For example, cryoEM enables the capture of high-resolution images of biological structures without the need for crystallization. This makes it possible to study molecules in their natural cellular environment. In particular, complex biological structures such as proteins and viruses can thus be analyzed.
We can actually look at these complexes in the context of a virus-infected cell. So this allows us to really understand mechanisms of virus infections, especially for these emerging viruses, and how to see how they actually enter cells, how they infect cells, and how they then reprogram these cells, and really see how cells are reshaped by these viruses.
Conducting research with pathogens that can cause serious disease requires properly secured laboratories. The BSL-4 lab at the cooperating Bernhard Nocht Institute for Tropical Medicine, one of only 4 of its kind in Germany, provides such a safe research environment. Scientists here are optimally protected against accidental infections, and this in turn effectively protects the community.
The biosafety level 4 laboratory, which we have here at the Institute, is a prerequisite to work with highly pathogenic viruses, including Ebola virus, Marburg virus, Lassa virus, Congo hemorrhagic fever virus. And we work on 3 topics. One is the understanding of the basic replication of these viruses. Second is to understand immunology and pathophysiology of these viruses in animal models. And the third aspect is to analyze samples coming from our partner sites in Africa from the clinical studies, which we analyze here in the BSL-4 lab with respect to immunological and other parameters.
Many highly pathogenic viruses arise endemically in Africa. The Bernhard Nocht Institute for Tropical Medicine, a participating institution of the CRC 1648, collaborates closely with the Irua Specialist Teaching Hospital in Edo State, Nigeria. This makes virological research on site possible. The scientists here are focusing on the Lassa virus in particular, a recurring deadly viral infection.
Lassa fever is one of the most important viral hemorrhagic fevers and emerging infections. And the challenge is that it occurs in sporadic epidemics in most parts in West Africa. However, at this specific site, the Irua Specialist Teaching Hospital, we see hundreds of patients every year. And so it's really a unique setting to study Lassa fever and viral hemorrhagic fever together with our partners from Nigeria.
Lassa fever is transmitted to humans via infected excretions of multi-mammat mice. Animals and humans are closely related and thus share health risks, including the risk of viral infections, and both depend on their environment. The living conditions of animals and humans cannot be considered separately from the climate in which they live.
We have collaborators in different areas. Here in Hamburg, we have a big climate cluster and climate research. We also have Institute for One Health, and we will also engage with these experts. So hopefully we can make a contribution by putting our piece of the puzzle into this whole picture.
However, the success of any research endeavour depends on the people conducting it, both today and in the future. The CRC 1648 has an integrated research training group in which international, highly motivated young researchers are prepared for future viral challenges. Not only the technical equipment of the CRC 1648 is excellent, so are its young scientists.
We bring a lot of expertise. We have international networks, we work with scientists abroad, and we have a great team. There's a lot of team spirit and people who are motivated to tackle this important problem.